All right, we're moving on to conditional specification. So another ridiculous figure, some sort of bar heat map thing on it. But this does show, so conditional specification is much more reliant on your environment for what kind of cells you become, right? So it occurs when the cell's fate is determined through interactions with neighboring cells and its position within the embryo. So fate depends on cell-to-cell signaling and positional information. So who's your neighbor? It's kind of keeping up with the Joneses, so to speak. So this development is regulative. So cells can compensate if parts of the embryo are removed, which is kind of cool. So if you take away a few cells, then the new cells that come into that location use their positional information to kind of take up the spot of what was lost. So if you isolate or transplant these cells, the fate is based on these new environmental cues. So it's predominant in vertebrate development, including mammals, and allows for developmental plasticity and this embryonic regulation. So it uses juxtacrine factors, which are cell-to-cell contacts, and paracrine factors, which are things that are secreted, like hormones, or mechanical stresses. So let's look at an example here. And so here we have a experiment where they took cells from one location, which are normal back cells, and from this blastula, and transplanted it to the region which normally would be belly cells. And so if there are if the cells from the back tissue, if they form back tissue, this is cells from the back tissue, so this is normal development, and this would be normal belly development. Let's go to the next slide here. So what they did is they took a, oh, sorry. Oh, duh. Okay. So, so let's go back. Okay, so in this experiment here, they took cells from this early blastula, and these cells are normally back cells, right? So you'd normally have these developing on the back of this tadpole or zebrafish, or whatever we got. I think it's tadpole. And so if you took those back cells and you transplanted them to the region that's normally belly cells, in conditional specification, you would get back cells growing on the belly, right? Because they predetermined an early stage based on these morphological morphogenic factors, right? But as you can see, the tadpole from the normal control, or the tadpole which would normally develop, would look like this, and the tadpole that did develop, where the cells were transplanted, looked exactly the same. And that's because even though these tissues were, or these cells were taken from the back tissue, the neighbors changed the fate of these cells and said, no, you're in our neighborhood, you need to be belly tissue and not back tissue. So it changes its cell fate based on its neighbors. What's also kind of cool is that in these, you can also like take a huge chunk out of this cell, and cells will grow to replace this. And if you were to do this, like, remember, if we would remove that B4-1, or I think it was B4.1 cell from the conditional specification for like that sea cucumber, that you would no longer have muscle tissue. But because of this conditional specification, cells will replicate, fill in this location, and you have normal development. You don't have to worry about a tadpole that, like in my picture here, I'm going to move myself over here so you guys can see this a little bit better. So you don't have like a headless situation going on where this tadpole is missing a head. Like the conditional specification will allow it to come in and fix that. So there's this guy, Weissman, who was into this developmental biology. He proposed this germplasm theory that states that each cell of the embryo would develop autonomously. And he proposed that the egg and sperm provided equal chromosomal contributions, which was correct, and that they carried an inherited potential for new organisms, which is also correct. However, he theorized that the chromosomes broke into chunks that were unique to each cell type, and that skin cells only had the part of the chromosome from skin cells, and that gametes were the only thing that had the full complement of material. So he thought that like your chromosome, I froze a second. Chromosome 1 had mostly like the top half of it was skin cells, and so the cells that were going to become skin, that chunk of the chromosome would break and go into that cell. So he had some good thoughts, but he had some very incorrect ones too. And this was hypothesized before Gregor Mendel did his stuff with genes. So here we have a test of Weissman's hypothesis. So there's this guy named Roux who did this experiment where he used a hot needle to kill off this half of this blastula very early. So that hot needle lysed this cell in half, so you only had half of an embryo from a very early age. And so this provided some credence to the autonomous specification theory that Weissman predicted. But then later, Hans Dietrich did another experiment and obtained different results that seemed to put that theory into question. So Dietrich isolated cells by shaking these cells apart. He later used seawater, so calcium-free seawater to pull them apart. So these cells then went on to develop into complete larvae themselves, regardless of missing the other cells. So in the previous experiment, he had half an embryo because that was model used autonomous specification, and then Dietrich did this experiment where theoretically he should end up with only 1/4 of an organism, right? But when he shook them apart, each one developed into its own organism. So this kind of goes with the idea of conditional specification. So this was the first set of experiments that proved neighbors were important in determining cell fate. By splitting cells up into 4, it resets the cells to the 1-cell stage, right? So as soon as at the 4-cell stage, if you break them apart into 4th, they just start over like they're 1 cell again. So he also did this pressure plate experiment. So he wanted to determine if reshuffling the cells and their containing nuclei would cause the formation of a weird mixed up organism. So remember, they're like in this 3D shape, right? So he wanted to know if you reordered them and put them all in different places, if you'd have an organism with an eye coming out of its back and an arm coming out of its head and things like that, right? So the way he did this is he put this developing embryo in between, or blastocyst, in between glass plates, so that, remember, there's 3 axes for human cleavage, but 2 for some of these other species that we're talking about. So it restricted one of those axes, right? So it could only develop on a certain way because that plate is in the way from allowing it to go in that direction. And so you end up, instead of having this normal orientation of your 16-cell stage, you have this completely messed up top and bottom are mixed up. So the animal pole, the top, and the vegetal pole, the bottom, are all shuffled because of this pressure plate, right? So this resulted in a mixture of the orientation of these cells, and so based on Weissman's chromosomal chunk theory, right, where each one of these, this would have the chunk of the chromosome for hair, and this or for skin, and this would have a chunk of a chromosome for muscle, yada yada yada. So if that were the case, we'd have this weird messed up organism, right, where, like I said, with an arm coming out of its head or something like that. But what happens is it develops normally, right? So this goes back to the idea that in conditional specification, it's who your neighbors are, and everyone has a full complement in the genome, and it's more based on selecting what genes are transcribed and translated into proteins based on that information of who your neighbor is and what you're supposed to be in that regard. And then, so this is just a quick comparison of autonomous versus conditional. So autonomous, remember that was the one from the last lecture that your state's pretty well determined from the beginning. That's determined by these proteins and mRNA inside the cytoplasm. It usually happens really early, before or during cleavage. You have very limited plasticity, in meaning that your fate is pretty much sealed from the beginning, and develops according to the predetermined fate, so you can't really change it. And then this is like an example would be C. elegans. And then conditional is what we just talked about. So external signals like your neighbors can occur throughout development, so early or late in development. So the plasticity is high, so you can change things, like when we did that transplant experiment for the back of the tadpole to the stomach of the tadpole. It's very flexible. If you isolate these cells, you may fail to differentiate or change fate, right? So because you have no neighbors. So if you isolate them, that information of what they should be is lost, because if you have no neighbor, then you're like, oh crap, what do? I don't know. No one's here to tell me what to become. And so an example of this would be the mammalian inner cell mass. So that is it for conditional. We're going to, in the last one for this section, we're going to quick talk about syncytial, which is with insects. It's kind of a different beast altogether. But then after that, we are going to continue on to the next unit. So I will catch you in the next one.